Welcome
Welcome
to
to
iGEM.
iGEM.
We just did that. Okay.
Our winter 2024 info session.
Woo. Thank you guys so much for coming.
Yeah, we also have this recorded, so it's going to be posted eventually somewhere. And yeah, so if you have any friends who aren't here, then you can potentially look at that. Okay, so what is iGEM? So iGEM stands for International Genetically Engineered Machine. Essentially, we're a big competition, the iGEM competition, and we do synbio projects. So we find a problem in the world that exists, and we come up with a solution that currently does not exist, and we use synbio to solve it. That's essentially the big iGEM picture, and that is the big competition called the Grand Jamboree that's held at the end of the year. So our team specifically at Waterloo, we are 3 sub-teams. So the lab and design, math and modeling, and human practices. And together we, as 3 sub-teams, come together to solve one problem. And throughout the entire year, from January to December, we use synbio to kind of design a project. And we present it at the international stage, which is the big competition called the Grand Jamboree.
So my name is Sasha. I'm the HP or Human Practices lead. I just finished my 3rd year in nanotechnology engineering, and I'm currently on co-op.
Hey guys, I'm married. I'm in my 3A term, biomedical engineering. Me and Yash are going to be co-leading the math and modeling sub-team. I'm also maintaining the website. I'll just do some other stuff as it comes, but yeah.
Yeah, so my name's Yash. I'm also a math and modeling co-lead. And I'm starting my free next term.
Hi everyone, my name is Mahnur. I am going to be the wet lab or the lab admin lead. I am in my 3A computational chemistry term, so excited.
Hi everyone, I'm Emma. I am in 3B biochemistry, just came off of an 8-month co-op, and I am the lab project lead. So that means I'll be doing a lot of the weekly lab meetings and managing research and project progression.
Hello, I know some faces from, I don't know where, but I'm Bethany. I'm also in the same position as Emma, just finished an 8-month co-op. So I'm also in 3B biochemistry. I am part of lab and design as well, so the 3 of us are lab and design leads. But specifically, I'm also the team admin, so I'm more in charge of the overall team structure and the administration of things like that.
Before we begin with the slide, we just want to ask you guys a simple question. What is synthetic biology? Like, what comes to mind when you hear synthetic biology? We'll just take answers from anyone.
Yeah, so synbio is something that's not really taught in a lot of biology courses here at Waterloo. We don't really have a faculty department, unlike McGill, because they're fancy, but we don't really teach synbio at all because, well, it's not really tied into something specific, but a lot of tools and techniques come into it. So out of curiosity, what do you guys think? Oh my goodness.
Sorry, do you mind repeating that? Online?
Oh, no, that was me.
Oh, okay.
That was my echo.
That was an
echo.
Oh, okay. If someone has an answer online, you can feel free to try and tell us your ideas as well. But how many of people, maybe if you're shy to like actually give an answer, like raise your hand if you know what synbio is, or you sort of know what synbio is. I see some like heads shaking.
Have you ever heard of it in class or anything?
And like,
It's not really like mentioned in class.
okay,
Yeah.
so clearly a lot of you don't know that much about it. I see a lot of shrugging and I have no clue faces, but that's okay. So really what synthetic biology essentially is engineering through genetics living things to create novel systems or products. And in iGEM, that can really mean a diverse range of things in terms of the tools and techniques you work with, but we work with various different types of molecular biology techniques, bioinformatics, and international standards for synthetic biology like BioBricks.
Yeah, so if you think of things like plasmid, genetic engineering, and even the word synthesize, making something that isn't really naturally there, that's where we tie in the biology with it. So think of like, yeah, plasmids, BioBricks, CRISPR, and those are all things that are tools that we can use for SynBio, and we bring in those different things, and well, there's a whole bunch of other bio things related, like bioinformatics, biostats, and that's essentially what our team does. We make something that doesn't exist. Yeah, so essentially a big part of our team, again, is that big competition. It's happening October or November. They kind of announce the dates. But the big competition is called Grand Jamboree. And in the past 2 years, it was held in Paris, France. Ooh. So hopefully we, go there again this year. We don't actually know if it's going to be held there again, but I think it probably is. So I think it was held Place de Versailles. Was that the name of it? Yeah. So in 2022, we had our Pinean Express project, and we had about 10 people go to present our project. And then this year we had 4 people go. Emma's one of them. So if you have any questions about like her experience.
Yash as well.
Oh yeah, Yash is online as well, and he also went. So I guess that was a pretty cool experience. Yeah. So previously the competition was actually held in Boston, and because it's because like before COVID they were trying to like. Well, it was held in Boston because MIT was the first university to kind of have the iGEM competition. And then they also have judges at the competition, and that's where all the international teams go to participate. Yeah, more recently they've been trying to like reach out to like more European side things, so that's why it's been held in Paris, France.
Yeah, it's close to 400 teams globally. It's a lot of.
Yeah. And then I guess more locally, so we kind of throughout the year, not just October and November, we also go to different conferences as well as different what we call mini jamborees. And there you get to meet a whole bunch of other teams. So for example, iGEM Guelph, Toronto, McGill, and then that's essentially a lot of Canadian teams. We get to meet them, meet cool people, learn about their projects as well. In 2022, we went to, it was called In Vitro 2022, and it was held by Concordia University in Montreal. And there was like a whole bunch of speakers, keynote people, yeah, a lot of like startup industry people as well. You get to like network. And then this year we went to a more biotech conference. It was also in Montreal, and it was held by McGill University. And we presented our project there. We got judged a little bit, and also, yeah, get to meet cool people. And if I remember, also it was like kind of like keynote speakers and things like that. So there was like a guy who was like on the round table of like talking with Biden about in vitro fertilization. So that was pretty cool.
Industry leaders
yeah,
in Canada.
Yeah, that too, and a lot of like professors across Canada. So we got to really network. And then I started talking to this guy who was like a really professor guy, and I didn't know where he came from, but that was pretty cool. You get to meet people. So there's a lot of conferences and a lot of different things that we kind of try to reach out to learn more about essentially SynBio across the world.
And as Bethany has been saying, it's a great way to network and collab with other universities. So this year, we actually hosted our very first iGEM conference in Waterloo. So we invited iGEM's Toronto and Guelph to join us here, and we had some industry professionals and also professors presenting, and it was a great networking opportunity with other universities. And in the past, we've also done virtual events with iGEM Copenhagen and other teams across the world. So again, great networking opportunity and a great way to meet people in the industry.
So a little bit of our team structure. This looks very complicated, but don't think of it as complicated. So our whole team, again, in Waterloo here, we work on one project. We're divided into the 3 sub-teams, so lab and design, math and modeling, human practices. Each sub-team is led by like leads. So we are the leads here, and Yash as well online. And also, for you guys would be like applying for sub-team members, which would be under like one of the 3 sub-teams. We also have other positions which will be discussed at the end of the presentation. So that's finance, wiki design, and social media coordinators. They will also fall under a sub-team, but we'll just explain that at the end. And then lastly, at the very top are our advisors. So we have a lot of profs. Our PI is Dr. Brian Ingalls. We have, off the top of my head, I remember we have Dr. Bar Moffat, we have Dr. Trevor Charles, who are from Department of Biology, and we go to them for a lot of genetic engineering like questions, sometimes dumb ones, but yeah. And then we also have, I'm pretty sure, like a lot of chemical engineering and other engineering profs. So off the top of my head, like Dr. Mark O'Coin and Dr. Valerie Ward. So yeah, just a lot of different advisors, people who are smarter than us and know a little bit better. But yeah, if you ever heard of those names, maybe they've also mentioned iGEM before. And of course, people with money, because that's always important, especially if you go to Paris.
Okay, now a little bit about our project last year. How many of you have maybe read a little bit or seen a bit of some blurb about the project last year?
We worked really hard on it.
Thank you. Thank you.
Thank you.
So I'll just do, even if you've familiarized yourself with it, I'll just do a new summary just to help you understand what we do in a year. Last year, we focused our project on tomatoes, but let's backtrack from tomatoes. And do you guys remember the lettuce shortage that happened just over a year ago? You
Yeah.
go into Burger King and they charge you extra for asking for extra lettuce. It's crazy. Anyway, that was caused by a problem virus called INSV. It's spread by insects called thrips from plant to plant, and it was causing wilting of plants, destruction of crops, and there wasn't a simple solution. You couldn't just simply genetically engineer the plants themselves to deal with this. And farmers, to deal with this, would sometimes have to burn crops to help eliminate the virus, or just use pesticides in general to prevent the thrips. But we all know that's not the most sustainable solution, so we thought we could do something better. Now, upon doing more research, we realized that this was a bigger problem of insect-vectored viruses that infect plants, including tomatoes, and we ended up focusing our project on TSWV, or tomato wilted spotted virus, which is a cousin of the lettuce virus, but it was just a bit more research, so that's why we focused on it. Now, to find a solution, we got really deep into the nitty-gritty world of plant immunology. Beth and I spent a lot of our
Because they were taking about
Reading.
341 immunology.
yeah.
No? Yeah, you did? Okay, me too. And I got confused, okay? There was so much immunology related to plants that we dived into, but like to break down our project, as Emma was saying, like it was just a lot, but that's what made our project really fun, because there's a lot of research that you can go into. Yeah.
Anyway, after that deep dive and a lot of twists and turns in our research, we settled on a solution of double-stranded RNA as a bit of a vaccine for this virus on plants. Now, this double-stranded RNA is a little bit like the RNA vaccines you've maybe heard of for COVID, except different molecular pathways. However, we wanted to push beyond that solution and make something more novel, because something like a spray vaccine, you just have to spray it, and then, that would treat a field, but that doesn't really treat the broader problem. So we came up with the idea of a living vaccine, using the concept of Pseudomonas syringae, a bacteria native to plant surfaces and the thrips themselves, to be a living, lasting, potentially spreadable vaccine for a concept of spreadable immunity. Then we delved into more details, like what bacteria we could use for our proof of concept, and we had to research, secretion, delivery mechanisms, and finally we ended up with a result of using E. coli in the lab as our proof of concept. We successfully genetically engineered it for the delivery system for that double-stranded RNA, so we're very proud of that.
Okay, so I'm gonna go into just a little overview, because Emma still talked about like the whole project and stuff. So what the lab and design specifically kind of did, like an overview, we developed like the conceptual Pseudomonas syringae based vaccine system for like insect vectored plant viruses. We also demonstrated successful like genetic engineering, and we also like provided the proof of our concept in the organism like E. coli to support the RNAi delivery.
For math and modeling, I'll just give like an overview. Me and Yash will go over it in more detail later, but it does come down to developing models for several things, one of which would include the bacterial dynamics of this proof of concept E. coli, the viral dynamics of TSWV, plant health and susceptibility, and as listed here, like modeling the RNA interference mechanism, including like its delivery and how it impacts the plant in particular.
The judges this year internationally said they were very impressed by our models. So yeah,
wow.
Good job to the math and modeling team.
What was interesting about our project, which was very RNAi based, was that we didn't win. We won bronze, but the top winning competition or like teams at the Grand Jamboree competitions, a lot of them actually focused on RNAi. So kudos on us for like being on track as to something that's up and coming in a lot of the agricultural like world.
Yeah, I think it was the top agricultural team, the top bioremediation team, and the top overgrad team had honestly very similar concepts to ours. They probably just had more money to maybe execute a bit further down the line. But go us. We're proud of our concept and we have plans to go further this coming year. Not with this project, different project, but we're setting our hopes high.
And for human practices, I'll go a bit more into detail about what exactly that is, but we got to talk to industry stakeholders directly about how this virus affected their plant growth, or we talked to someone who works at Loblaws for procurement about how it affected their inventory and their stocks related to these plants. And we made sure that our project was completely ethically aligned and addressed any societal concerns, as well as fostering awareness and acceptance through effective communication, whether that was sharing extra Instagram posts, making sure everyone's aware of our project, why we're doing it, or just with the community engagement with hosting that conference so that other universities know what we're up to and we can all work together.
So we just talked about last year's project. Here are some projects from before that. Again, I'm just going to summarize them, but you can visit our website and you can learn a lot more about each of these projects, including projects before these ones. But just really quickly, Remind, essentially, they created this bioreactor. Within this bioreactor, they had proteins that these proteins attracted and essentially removed heavy metals from wastewater that comes from electronics manufacturing. Neurodetect was essentially just like a tool for diagnosing ADHD, and they would do so by measuring biomarkers in the patient's urine. And then last year, Pinene Express, they bioengineered their own yeast. And this yeast essentially made the main compounds of cannabis. And this would just be like a sustainable alternative to just cultivating the cannabis. So.
Yeah, and we made weed. Yeah, for medical uses, and we had a good.
But we made weed.
But we made weed. Okay.
Plot twist. Whoa.
Oh, can you code this?
Just refresh the page.
Okay.
Maybe the weed part makes that happen.
No, this was the best slide. It's okay, guys. A
I
little
feel
technical
like
difficulties.
I trust that having survived COVID, you guys are used to dealing with technical difficulties during online calls.
You know, I think I know what the slide. It's the question slide, right?
Yeah.
Yeah. Okay. So while
Do you guys have questions?
yeah, any questions before we dive into more specific roles and responsibilities of each sub team so far?
Anyone from the?
Also, if anyone online wants to either speak or write something in the chat and then Yash speaks for you, you can go ahead now and ask any questions.
Is everything making sense? Yes. Nice.
Yes.
Maybe if we just see,
Yeah.
we'll wait a minute in case people type. Where are you guys from? Which faculty? Hands up if you're science.
We got a question.
We got it.
Sorry.
Cool. Yeah. How about okay, down engineering. Nice. Okay, down math. Nice. Okay, arts.
Business?
That's not a
You know-
faculty.
Oh, right. True. Laurier students intruding our conference.
No, I don't think so.
Any other faculties I didn't mention?
Science?
We're good? Okay.
Wait, science.
So
I did, bro. That's the
Yeah,
first one.
iGEM could be anyone. We're not exclusive. All the sub teams were open to anyone, and excited to looking at your skill set. But a question online.
So the first one is, do you specialize in one domain of biology, example, human biology, plant biology, etc.?
This is important.
No. So as I mentioned in the beginning, which I will emphasize at the end, we're finding a problem that exists in the world and making a solution. That's essentially what we do, and we use SymBio. So that could be any problem.
And it really depends on the interest of the team we select. So right now we're in the process of team selection. We're gonna find our team, and then we're gonna learn what you guys are gonna be interested in, and we're excited to see what you guys bring to the table. The fact that the past 2 years both had something to do with plants is by no means a limiting factor to plants. We're really open to anything. Using bacteria in the lab is pretty expected for iGEM, but beyond that, our actual concept can really be anything.
Okay, next question is, what is project selection process like?
Okay,
Do we want to get into that now, or
so
it's kind of?
We have a slide on that, don't we?
Yeah.
We do? No, I don't know.
Okay, well, so right now, I trust that all of you guys have seen the link to the Google form for online applications, correct? So those are due on the 16th, I believe.
Next Tuesday.
So we want you guys to fill those out, encourage other people who you think might be good potential members to fill those out. Shortly after that, we will look through all those applications, select a bunch of candidates, and we'll send out emails to some individuals that we'd like to interview. And then we aim to have the team finalized by January. This isn't the exact same timeline as the previous year because everything changes with the timing of lead handover. But the plan is to have the final team by end of January. There any specific
So, like,
question?
for
sorry,
project
the question
selection,
was project selection.
okay, yeah. For project selection specifically, so essentially once we have the team, within the first month and a little bit, we kind of get into groups of different people. So we mix some labs, some math people, and some human practices, different programs, different interests. We ask you what your interests are, and we will kind of list out the different topics that you can fall under. Essentially, the possibilities are endless, though the topics are like very general. And you kind of make up a kind of topic within your group of like 5 or so people, and that's how we kind of think of a project. And then we kind of present it to the whole team wide, and then afterwards we kind of vote on a project that we all like. And then after that, if we all like it, then we kind of continue with the research into it and go into more depth with that topic. So for example, the garden, for example, started off with a really general idea, and we kind of presented to the whole team. Everyone kind of liked it, so we went with the idea and went into more depth into it.
Yeah, and also finalizing project selection, often we go to our advisors to help bounce around ideas to see what the professors have to say. And then we, go back to the drawing board as needed, revise, finalize our idea, and boom.
Yeah, some people join iGEM with an idea already in their head, which is great, but also most of us don't, and we don't even know what to do. So we kind of look around, search up online if there's current problems in the world. That's where the lettuce idea came from. I was like, what are some current problems in the world? And
Eating
it
a
says
sandwich,
lettuce.
right?
I was eating a sandwich and I said lettuce. So yeah, we kind of come up with ideas kind of randomly.
Okay, so let's get more into lab and design. So what is our role? We design biological systems, genetic circuits, proteins, really depends on the specific problem, very often plasmids, to address a real world problem. Now, obviously the whole team is involved in project selection, but lab and design is really involved in the nitty gritty of the initial ideation stage to bridge the project selection stage to the actual execution stage. Because we're the ones who are going to have to be doing it in the lab. In the lab, we may cultivate bacteria, plants, or other species relevant to the project, and we carry out and troubleshoot molecular biology experiments in order to gather experimental data. And this data is essentially an aim to prove that, make a proof of concept and establish that our idea is a good one.
Yeah, it's always cool going to the competition and actually being able to present what we have in pictures, or like actually bringing something like physical. We're like, here you go, we have weed, but like that's the cool part, and we actually get to make it.
So what can you expect to be doing? When I walked into iGEM last year, I didn't know what I was going to be doing, but now I have like a nice little sentence. So you're going to just conduct a lot of literature research, like a lot of research for proper design of experiment setup. You're also going to like utilize like gene editing software such as like SnapGene, Benchling. Don't worry, we have so many like tutorials on that for you guys, so don't worry. There's also going to be like some like in-lab training, but we'll get more into that like when the team is finalized. And it's also like in general going to depend a lot upon your like expertise. So if you're really good at bio, or like if you're really good at chem, or like if you're an engineer in lab and design, like you're going to be like utilizing all of your skills to like your full potential. So
yeah, we also expect probably to do the most of the wet lab stuff in the summer. So if you're on campus or doing co-op in Waterloo in the summer, that's really great. If you have extra time to spend in the lab. But I spent a lot of time out of the lab. I just came off of an 8-month co-op, so I found myself more in the research and ideation role, but your specific role can really depend on what you're busy with and what expertise you bring to the table. Okay, so a bit more of our design process. This really varies a lot every year because the design is not as simple as pick a plasmid, finish the plasmid, boom, that's it. No, there's a lot more to it. So I'm going to go back to this past year's project just to discuss this a little bit. The first stage of design process, which lab had to deal with in terms of from a research perspective, was, as I said, go really deep into plant immunology and consider, okay, what does our biochemical solution to this actually look like? We looked into proteins, other chemicals. In the end, we came up with RNA due to some proven efficacy, and we wanted a lasting effect. Then we combine our concept and then also practicality considerations to choose an organism to design systems around. So we chose E. coli because it was similar to our concept organism, which was Pseudomonas syringae. But E. coli, I hope you guys have all heard of that, but that's very common in the lab, easy for us to use. And we had to choose that before we could go further into our design process because a lot depends on the organism you're working with. And then once we had the organism and our immune generating product chemical, we had to work out all the nitty-gritty details of how we deliver it, how are we going to ensure it's stable, how do we make sure this RNA folds properly. But all that nitty-gritty stuff, it's really going to depend each year, depending on the project, the organism, and our goal.
So a week in our silly little life in lab and design. So the sub-team meetings and the team-wide meetings, which will occur like probably like once, like obviously when the team is all set up, it'll happen like on the first day, and like later on it'll be probably like monthly or like maybe every like 3 weeks or something.
For team-wise, but
For team-wise, yeah, for team-wise. And it'll only consist of like 1 hour like team-wide meetings, and then for research and lab work, like in lab work or maybe like just researching, it could take around 3 hours, but like it's just like a relative number we're giving it right now because we don't really know like what our project's going to be next year or this year, and in total we're thinking of like probably like 5 to 10 hours per week. It could be less, it could be more, like depending on how like really good like the project ideation comes through, and work-life balance is important.
Yeah, we do spend roughly an hour a week in just lab and design meetings, and also, yeah, we aim for 5 to 10 hours. We're obviously flexible around things like midterms, but you should expect if you want to get something out of iGEM that you will have times where you sort of allocate, sometimes bigger blocks of time to really focus on something and get a task done, get some research done, and that's how we really get productive.
Yeah, I was also going to mention, like, for example, if you go to Paris, you go to Montreal, that's a whole weekend thing. So obviously you're going to spend more than 5 and 10 hours, like, there. Or, for example, things like running a gel and you come back an hour later, maybe in total you spent like 2-3 hours there already. And then you come back and then you kind of come back at 9 p.m. and then you leave and then you come back at 30 minutes later, check on it. Oh, it didn't run and you got to do another hour. So it varies, yes.
Yeah, hopefully we will have a nicely set up lab schedule that people can sign up and sort of plan for ahead of time. And this meme is like a bit of an exaggeration. It's not actually crazy nuts. It's just funny.
Well, we did stay up at 2 a.m. in Montreal.
Yeah.
So it depends.
But that's fun stuff as well,
Yeah.
Okay, what are we looking for in a lab and design member? That could be any of you, that could be any faculty, any year. We're really looking for a balanced team this year. We want someone who's eager and excited to learn, hardworking, self-motivated, adaptive. You can take initiative. Some technical skills, you don't need you don't need all the specific technical skills, but it's definitely a benefit if you're comfortable working safely in a lab setting. And it's good to be familiar with some basic techniques such as plasmid cloning, PCR, etc. Just to emphasize this, we really don't need prior experience, and you should be prepared for the fact that we're a student team. As leads, like none of us are experts in everything. So, it's not like as leads, we just assign very specific tasks. As a team, we're collectively learning everything together. So we want people who are prepared to join the experience of teaching ourselves as a team. And you know what? You could be a first year and we're stuck on a research hurdle. And you could be the one to be like, you know what, I'm going to spend a couple of hours trying to look into this, even though I find it super confusing. I don't really know what's going on. I'm maybe going to get my friend involved. And you could be the one to sort of solve a big problem for us. So, we really want those people who can take initiative and are ready to learn.
So Emma mentioned adaptive, and the reason why is because lab and design, I think the biggest part I've learned being on the team for this is my 3rd year, is being able to troubleshoot, because nothing is ever going to go as planned. You're going to design your best plasmid, looks beautiful, but it doesn't work. So it's happened 3 times, 4 times, so the idea is that you have to learn how to be adaptive, and whenever there's issues that come up, you kind of try to figure out if there's a way to solve it, if there's ways to troubleshoot it. And so I think that's the biggest part I've learned as being part of the team.
Yeah, and we want team players, but people who can also are comfortable taking on a test themselves as well.
Math and modeling, wow, okay.
They're the unquestionably
We're not
cool
gonna read that.
nerds with
Okay.
code. Sorry, I had to read it.
Fine, okay. Math modeling, yes. I would say these bullet points kind of describe it pretty well, develop mathematical models, predict behaviors, aid in design choices, and troubleshoot stuff that lab and design does. Me personally, I would say the core of it kind of does boil down to just making or doing really good research, meaningful research, and then converting our research into, depending on the project, either well-written source code or scripts, like a well-designed 2D, 3D model. It's not included on the slide, but even just molecular visualizations with specific software, like PyMol, Rosetta, that kind of thing. That kind of lends itself to these skills and what you would expect, writing scripts, Python, MATLAB. Doesn't really matter the language too much, more so that's kind of tends to be the case. We end up writing a lot of code, pushing that to GitHub, extracting data sets, driving ODEs, CAD modeling, etc. But again, the main focus or the main thing I want to drill, it does depend on the project. And some projects will include all of these, some projects will include some of it, but inevitably you're going to be learning something really technical and useful.
Yeah, so for Gordon, we split our work into different subsections, so bacterial dynamics, RNAi, SIR, and viral dynamics. And luckily for our subteam, we don't really need an extensive knowledge of synbio in order to model. So for Gordon, we mainly used a mix of differential equations and stochastic ones, basically, for different purposes, such as modeling the spread of a virus, bacterial growth, and E. coli population. And I guess we also went through a bunch of different papers to basically pick out which parameters and values would be our ideal candidates for generating the model. And honestly, I think one of the most fun things is to see how many different models exist and trying to see which one fits our project the best. And yeah. So here's an example of how we modeled RNAi for Gordon. So we mainly used a lot of different stochastic differential equations in order to run a Monte Carlo simulation. And it might look a little intimidating, but we also don't fully come up with these modeling equations by ourselves. So even if you don't feel familiar with the math theory that's being used, that's totally fine, because most of it will probably just come from research papers.
What Yash said, so don't get intimidated by this. This is a lot, but yeah, a lot of it is borrowed. A lot of it is sampled. We put equations together. What makes sense? We go through a pretty rigid thought process to get to these graphs and equations, writing the scripts to create these plots. So, and this goes to what I said before. For the 2021 project, they did some protein optimization modeling, and they used, I think this was PyMol, but this could be Rosetta. And then they used Onshape, a CAD software, and they made a physical design, as you can see there.
Yeah, so on a week-to-week basis, we'll probably be having one team-wide meeting where we discuss each team member's updates just to keep track of how the project is going, essentially. And otherwise, once we distribute the work amongst the micro teams, People will basically be collaborating in their own time to like read up on papers and do modeling work by themselves. And don't worry, because we encounter roadblocks very often, which is why we think working together to troubleshoot everything is like very important.
Okay, what we're looking for. Yes, technical skills, someone with a good, solid technical skill set. As we mentioned, like programming, scripting experience, CAD experience, really Python, MATLAB. Even if you're like a JavaScript guy, C#, C++, like if you've done programming, that would be amazing. CAD experience, SolidWorks, AutoCAD, Fusion 360, any of those. Same case if you've done CAD. As we've mentioned, research. As corny as it is, I do want to like stress, yeah, like soft skills, because yes, we more than anything really, like we are looking for someone with initiative, with a strong work ethic, good time management, and I think personally more than anything, like are committed and passionate, because I find if you care about this project, if you care about the team, if you care about it more than us, like everything else kind of lends itself. So like you're committed and passionate, and then you're willing to learn how to write Python scripts. You're willing to learn how to make a good 3D model. So really, we do care a lot about soft skills and a passion for synthetic biology and the project.
So human practices, I'm just going to go over a bit of what it is and what we do. So essentially, we ask a lot of questions that help make our project clear about like why we're doing it, who we're doing it for, and all of the kind of what's the story behind our project? Why are we even doing this? So to do that, we talk to a lot of stakeholders, which for Garden, it wasn't only just people like that work at Loblaws or grocery stores. It was also farmers to see how it affected their plants, but also just consumers to see if you saw an increase in pricing in tomatoes or other produce. So we got to talk to a lot of different people. And we also want to make sure our project is always ethical. So we got to look at the governing laws, all the patents, and if we ever do want to go to market with this project, what are the production and distribution costs? And also, like I said, what do people think of our project? Is it even needed in the real world? And if it is, how is it needed? Why is it needed? And we apply what we learn by making sure that we are always looking at public opinion and the experts. And how exactly do we do this? We send out a lot of emails. So in the beginning, when we have our project selected, we're going to basically focus on nailing down that stakeholder list so we can always contact them if we have any questions about our project and again, how it affected them. And also if they have any ideas of how to go about this, if there's anything specific in our project that we want to tackle for them. And again, we need a lot of creativity with coming up with that list of stakeholders and just how to market the project better and making sure it's out of the box and how coming up with ways that we can have it stand out amongst other competitors. And we were also in charge of like the GEM Start conference, so it was really great to reach out to those other universities. A week in the life, it's honestly pretty chill. I would say it's about 5-hour commitment every week. And as we said, that the team-wides aren't going to be happening every week, so they'll mostly happen in the beginning, but we will have one weekly sub-meeting for about an hour. Sometimes we don't need the full hour to be talking, but they will be the full hour just so it can be a work session for you to get any of your assigned tasks done. And outside of those meetings, you're going to be in charge of doing outreach with other people in the iGEM community or those stakeholders and reading any policies and documentation to make sure that our project is in line with Health Canada's requirements or FDA requirements as we need it. What we're looking for in an HPE member is someone that is willing to have fun and also work hard. Like I said, it's honestly a bit of a lower commitment team, but you do get out what you put into it. So if you're constantly volunteering and taking initiative, like things like GEM Start and also getting on calls one-on-one with people that are in industry or professors, that would be great. So we always want someone that is willing to take initiatives and someone that is solution-oriented and a critical thinker. So you can always come up with any extra solutions to any problems we come across.
Before I go into this, I just wanted to add something. So actually, no sub-team is actually more important than the other. We need all three equally. So for example, 2 years ago, we won the HP award for really good human practices and ethics. And then this year, we got a little bit applauded for our models for math and modeling. So our team is about, I think last year we had about 50 members in total, approximately like 7 to 10 sub-team members within each sub-team. And also a big interesting thing about iGEM is that actually some iGEM teams, their idea was so good that, well, not Waterloo, but it could work. So their idea was so good that it had a part 2. So part 1 was in like 1 year, but they couldn't finish the entire project in 1 year. So the second year was the part 2, and I forgot the name of it, but I think it was like a space-related food thing for like astronauts, and they actually became a startup afterwards. So iGEM always has things where it can lead to something more. Our team so far, we just tend to stick to 1-year things, but it could happen if we have a really good project. Okay, so for the 3 specific kind of additional positions that we're also hiring from, in the application, at the end of the application, it's there, and it'll ask you if you want to also apply for any of these 3. So these 3 positions are very important to us. First of all, finance. If we want to go Paris, we want to go on any trips, we always need to have someone organizing our finances. We get different scholarships and different like amounts of money and funds from different faculties, since we have science, engineering, math students. So it gets a little complicated, but we kind of need people to help us organize it. And
If you have creative fundraising ideas, please, we want to hear them. We want to see what you can bring to the table.
We did Krispy Kreme before and some auction things before as well, but obviously better fundraising ideas is always good. Our main wiki design. So wiki is essentially like the website that we have. Every year we make a wiki design. So every iGEM team every year makes a wiki. This happens around like September, October. But we need people who know how to code, because clearly I cannot, because I'm biochem. But UX, UI designers, anyone who knows how to make websites, those are very important for us, and also graphic designing with it, like throughout the year. And the last is social media. So our main form of communication is Instagram, Facebook, but we post a lot of our links and things like that there. So it would be cool if we had people who know a little bit more about how to like design cool graphics and things like that. So these 3 positions are, you can apply for it at the end of our application. We kind of do give a little bit of preference for people who have experience or would like to help us with any of these 3 positions. If you're in this position, say you're a finance person, your main role is to do the finance. However, you'll still also be under a sub-team. So for example, I'm also finance, but that's my main role and my main focus. But I will also be under lab and design and attend those lab and design weekly meetings, just so you are like aware of what's going on with our like the project. But you would be working mainly as that, like as finance, for example. So I guess this goes back into, I don't remember who asked, but one of the online questions about what can iGEM do? So kind of just to end off our presentation on like a cool note, but so iGEM, essentially, we have a lot of things called villages, and villages are essentially like topics. So some are like bioremediation, energy, agriculture. There's software, there's AI, nanobots. If you're more interested in hospital, like, or disease patients, like there's therapeutics design, diagnostics, there's pharmaceuticals, there's drugs. So our project within like the past 2 years have been agriculture and like environment sustainability, but our project doesn't have to be that. Our project can be pretty much anything and would fall under these villages. So if you're ever interested in what can become an iGEM project, you can look at their website. Like there's the main iGEM website, and you can probably scroll down, look at previous projects that have won in the past, and see what kind of project that they've done. Yeah, and just to end on a cool inspirational note, again, we're making something that doesn't exist, and that's solving a real world problem that people have. And we try to make something that is an impact onto the world. So yeah, how will you make history in synthetic biology?
Wow.
You look skeptical, but it might happen.
Yeah.
Thank you.
So thank you. Our application, I'm pretty sure you've seen the link everywhere, but our applications are already opened, and they're due on Tuesday, January 16th at 11:59. As always, if you already have any questions, you can always slide into our DMs or our email. We're always checking that. And then the links if you want it.
Any other questions that would be relevant for everyone to hear? That's also open to people online as well.
Someone's asking, when does the interview process begin?
That was from before.
Yeah, it didn't get answered.
Oh, okay.
I think we're looking at end of January.
So I think we want to do it sort of
next week.
Some of that is dependent on how many applications we actually get, and then when we're all available. But it's gonna happen before the end of January,
Yeah.
ideally. So, third, fourth weeks, and then hopefully we'll have a team finalized after that. We can't give exact dates because obviously we don't have, all the numbers yet. But yeah, as soon as possible, really, in our eyes. You?
So, how many people are usually behind those the final categories which you mentioned on finance?
Yeah, finance, social media,
Yes,
and
is it not just one person or is it team?
we're looking about like 2 to 3, perhaps we're about 2 for each of those.
Yeah. Some of these don't have complete precedent the way we want to run things this year. We want more specialized people this year, but also how invested you get into the other sub-team you're involved with. That's up to you. So don't be limited by the fact of, but I still want to do a lot of lab and design. If you are able to do a lot of lab and design, we also welcome that. You just, if you apply for that, just know that we're expecting some initiative and like a bit of extra time put in for those roles if you sign up for them, because that is going to be your responsibility within, a small group or committee.
Actually, specifically for wiki design, just going back to your question, we look about for like 4 people just because that's a big task in itself. I think social media maybe like 2, and then finance maybe 2 to 3.
I think we're more willing to have more for wiki because the website's like so important.
It's a lot of work when it comes to crunch time in September, October, and that's a big part of our project. So wiki design team in itself, we might have hiring sessions afterwards as well.
Yep.
Yeah, so it was mentioned that like the math and modeling sub-team, there were like micro team meetings. So like, what are the micro teams within the math and modeling sub-team?
Depending on the project. So assuming it was a project where we would need some Python scripts and we would need some CAD models, micro team specifically dedicated to just doing the coding, micro team specific to just modeling. But then even then, it's like you can easily switch between micro teams if you wanted to tackle on both. But yeah, it depends on the project. So it's hard.
The whole team tries to we try and go along and work as it makes sense for the project we're working on to just try and achieve our goals with the people we have.
Yeah. And even like, say you're a lab and design member and you're really interested in knowing what math and modeling is doing, feel free, jump in their meetings and just listen on. Even the same thing as some person who math and modeling who practices want to come into the lab, see what is going on, you can always do that as well.
And then we have a question, how many new team members are you taking in each sub-team? I don't think we're limiting it to any specific number. If you stand out, then we'll be happy to take you. I think in the past we've done about 8 to 12-ish in per team, but there's no specific number that, okay, we only have 12 openings and we're only going to take 12 people. We can always take more, we can always take less. So there's no specific set kind of quota that we have.
Being in Waterloo, because of co-op, we tend to err on the higher side just because not everyone can be in person. So we just need to make sure we have enough people to do stuff. But we really just need people who can imagine themselves getting invested in this and, caring about the project, putting effort into it, and building the team around them.
What's the interview like?
It's really short.
You'll get some details in the email. So for example, for lab and design, there might be just a bit of reading we might give you as prep. You can the Google form application is sort of a good idea of the type of questions we might ask. We want to get to know you a bit more. We want to know a bit more about your responses, and you'll find out
your
more
interests too. Like, let's say you already know something you want to work on, that's cool too. Yeah. So we're essentially done our presentation. We're gonna actually stay here behind to answer any questions if you ever have any. Is there any other questions online?
Not yet.
Not as of yet.
Okay, yeah.
If anyone wants to ask me last minute questions.
Coming.
Thank you.